All right, so we're going to talk about the second half of photosynthesis, which can be called the light-independent reactions or the Calvin cycle. We really don't use the term the dark reaction anymore, but the second set of reactions, so we had first the light-dependent reactions where we made ATP and NADPH, and now in this second set of reactions, we're going to use those products from the light reaction to make sugar. It does happen in the stroma, where the light-dependent reactions happened on the thylakoid membranes. The Calvin cycle takes place out in the fluid or the stroma, and it is just a series of chemical reactions, whereas the light-dependent reaction had structures and membranes and proteins involved. This is basically one chemical reaction after another. And the purpose of it is to take our carbon dioxide that the plant has brought in through the stomata, right? The CO2 goes in and it finds its way to a photosynthetic cell in the mesophyll, and then it makes its way into the chloroplast and it's dissolved in the stroma. And so we take that, the plants take that carbon dioxide and they use the energy from ATP and other high energy products like the hydrogen from NADPH and make sugar, or glucose, which is a simple sugar. So this graph or this diagram is looks a little bit more complicated than what it is, but CO2 enters the cycle, or it's dissolved in the stroma, and when it's in the stroma, there is also another compound called RUBP, which we see right over here, RUBP. It is a 5-carbon compound that picks up CO2. So it's also known as a carbon dioxide acceptor. So we call this process carbon fixation. When those two things join together, it forms this really unstable 6C compound. It doesn't last long at all. It doesn't even have a name. That's how quickly it breaks apart into 2 molecules of PGA. That PGA gets converted using energy from ATP and hydrogen from NADPH, and that forms pGal. So the pGal is now what's going to then become glucose. So the pGal that we form here, and I'm not worried about how many or whatever, but just know that we make a lot of this. The plants make a lot of this. And so 2 of those molecules can exit the cycle to become glucose, and the rest of it go back to make more RUBP. So the carbon cycle, or the Calvin cycle, you might want to stop and rewind and listen to that explanation again. Now, to put it into words, right, we know that carbon dioxide comes in through the stomata, through the pores that we looked at in lab, right, the stomata. The carbon of the carbon dioxide is combined with RUBP, like we said on the diagram, and when RUBP and CO2 join together, we call that carbon fixation. This forms a super unstable 6C compound that will break apart real fast, so quickly that it doesn't even have a name, and will break apart into 2 molecules of PGA. Now, PGA is an intermediate of this process, and what I'm telling you is a greatly pared down version of a lot of chemical reactions. So when CO2 and RUBP join together and then they form PGA, now PGA will be converted into pGal using energy from ATP and the hydrogen from NADPH because it's energized, right? That electron was energized by photosystem 1 and it's carried by the hydrogen of NADPH. So that energy and that energized electron will convert PGA into PGAL. PGAL basically has two routes. Either it's going to be combined to form glucose, 2 of those will be combined to form glucose, or the rest of it then that doesn't go to make glucose will be reconverted back into RUBP because you have to have RUBP there to be the carbon dioxide acceptor. So you have to start with it and you have to end with it, okay? Now, plants have a problem because of their stomata, right? The stomata has to be open for carbon dioxide to come in and oxygen to go out, but like we talked about, that's going to be a big water loss dehydration problem. So because of that, because their stomata have to be open so that the CO2 can come in because otherwise they can't do photosynthesis, that is going to allow water to escape the stomata the entire time it's open. So plants have had to make some trade-offs between how much photosynthesis is happening and how do they prevent excessive water loss, because to leave their stomata open all the time to get as much CO2 in as they can during the day, well, they're going to lose a lot of water. So what a lot of plants do is they'll close their stomata when it gets super hot and dry, and then that will prevent water loss, but of course it also slows down photosynthesis. So there are definitely trade-offs. Now, other things that can affect the rate of photosynthesis would be, like we were kind of just talking about there, water. You have to have water for the light-dependent reactions. We know water comes in from the roots, right? It goes out the stomata, but it comes in the roots. It's a kind of a one-way path from roots up the plant and out the stomata. And if there isn't any water in the ground, well, therefore, then there's not going to be any water for photosynthesis to take place. So plants try to slow down the water loss by covering their leaf surfaces with that waxed cuticle. Some leaves have a very thick wax, some a little bit thinner, just kind of depends on where they live. But that is one way that plants have evolved a mechanism to kind of slow down water loss. Another factor is temperature, and photosynthesis doesn't exist if there aren't enzymes. Enzymes control every step of every chemical reaction that occurs, and so they need enzymes. Enzymes don't work very well at very extreme temperatures, so they work the best, above freezing and below super hot, right? So in the Fahrenheit world, that would be, above freezing, in the 40 degrees Celsius range up to about 85. If you go below it or you go above it, the enzymes could be damaged and then they just don't work right. So if the enzymes aren't working right, then they are not, then photosynthesis can't take place either. And then, of course, the last factor that affects photosynthesis is light intensity. Clearly, the more light you have, the faster photosynthesis goes. And, there really isn't a capping on that. I mean, as much light as possible is good for the plant. Of course, on Earth, when you have high light intensity, you usually have high heat, which does cause a problem. So anyway, light intensity it does increase the rate of photosynthesis.